So it's a rather curious title, and I hope it'll make some sense as I go through it. But before I start, I'd just like to point out that this talk is co-authored by Lucia Guther, who's my long-term collaborator for 77 years and life partner. And that's a little bit like these characters here, Alice and C.C. Wang, who are, as Meg said, pioneers of molecular parasitology and also life partners as well as professional partners. And so I'm very, very honored to receive this award for several reasons. Firstly, because I've been selected by an esteemed peer group, but particularly because it's sponsored and bears the name of Alice and C.C. Wang. Their joint work on Giardia, its metabolism, cell biology, its RNA viruses, and more is the stuff of legend, and suffice to say that our collective knowledge of the Giardia parasite, would be primitive without their contributions. And as if this wasn't enough, CC also devised and directed an outstanding research team into African trypanosomiasis for 3 decades, covering discoveries in glycosome composition and biogenesis, drug modes of action and trypanosome chemotherapy, and trypanosome differentiation and cell cycle control, and being really one of the earliest adopters of rigorous analysis of kinases, cyclins, centrins, and other molecular components that coordinate parasite cytokinesis and mitosis. He also did brilliant work on the biochemistry of the trypanosome proteasome, which is very, very poignant at the moment because it's currently a very sought-after target for antiprotozoal chemotherapy. So, fortunately, he never worked on the cell surface. So it leaves a small window of opportunity for some of the rest of us to make some kind of contribution. So anyway, it's a real honor to have an award bearing your names. Thank you so much. Okay, so we do work on parasite cell surfaces. We work on kinetoplastid parasites primarily. And so this shows little cartoons of the cell surfaces of the African trypanosome, the Leishmania parasite, and Malcolm McConville, who's speaking today, has had a lot to do with this one, and Trypanosoma cruzi, the South American cousin. Over the years, we've worked on isolating and characterizing structurally and functionally the cell surface molecules of these organisms. Today, I'm just going to focus on one, and that's the African trypanosome, Trypanosoma brucei, which is an entirely extracellular parasite, unlike the others, doesn't invade host cells, and it causes human African sleeping sickness, which is a truly horrible disease, and also related cattle diseases. Just to point out that it exists in multiple life cycle stages. So this would be a cartoon representation of the cell surface of the bloodstream form of the parasite, bearing this major surface glycoprotein known as the variant surface glycoprotein, characterized by my postdoctoral mentor George Cross. And then when it's in the insect, it differentiates into a biochemically and morphologically distinct form of the parasite called the procyclic form, and has a very, very different cell surface made up of these spiky polyanionic type of molecules. Now, we study glycosylation in trypanosomes, so why would you do that? Well, it's a genetically tractable eukaryote with both common and unique features of eukaryotic protein glycosylation. And that means the biology of the organism makes it really an optimal model organism to study certain types of post-glycosylation PTMs. And at the same time, the difference between itself and its host, that's man and cattle, may provide opportunities for therapeutic intervention. So it's got both of these nice attributes. So I'm going to start with briefly telling you about one of these common pathways to all eukaryotes where its underlying biology made it a very good model organism. And that is actually in the area of GPI, glycosyl phosphatidylinositol. That's why we're going to call them GPIs from now on, membrane anchors. And this is a textbook picture of how membrane proteins can be associated with the lipid bilayer. Obviously, they can span once, they can span several times, they can be peripheral, in other words, physically associated with something else that spans the bilayer, or they can be lipidated. And some of those are lipidated and attached to the inner leaflet of the plasma membrane. But this class are always on the outer leaflet of the plasma membrane, and it's through a covalent linkage to this complex glycophospholipid or GPI structure that enables that physical association between those proteins and the surface. Now, this is ubiquitous amongst the eukaryotes, but it was really discovered and delineated in the African trypanosome, not because the people who work on African trypanosomes are particularly smart, although we're not too dumb either, but because it's the world's most natural overexpressor of GPI structures. And the reason is that if you were to take a little bit of plasma membrane off the trypanosome and make a cartoon of it, of 20 nanometers square by 5 nanometers thick, you can see that it's covered with this incredibly dense palisade of identical variant surface glycoprotein molecules, each of which are a homodimer, but on each monomer there is a covalently attached GPI membrane anchor. This means that we were actually able, in the early days, to purify literally grams of VSG to get enough material to perform the various analytical experiments necessary to delineate its structure. So it played an important role in that particular field. Now, interesting though GPI structure determination was, the key question is, can we exploit GPI biosynthesis in the parasite as a potential therapeutic target? And this is a highly simplified image of how GPI biosynthesis works. This was delineated by the labs of Paul England and George Cross initially, and they developed the cell-free systems which enable you to study this process. And broadly speaking, you start with phosphatidyl'inositol, you add N-acetylglucosamine, you deacetylate it, it flips into the inner surface of the ER, it gets 3 mannose residues added and then an ethanolamine phosphate, and it's good to go to be attached to the protein. It gets exchanged for a C-terminal peptide. So that's fine, but how conserved is that pathway between the parasite and the host? And one of our early indications that there were significant differences goes back to 1995, actually work that Lucia did, where we found that this pink reaction strictly precedes the blue reaction in T. brucei, whereas in humans, the blue reaction strictly precedes the pink one. And I'm not going to bore you with the details of exactly what those reactions are, but that told us that there's a fundamental difference in substrate specificity of these early steps of the GPI pathway. Now, unfortunately, you can't go to Sigma and buy substrates for a newly discovered pathway, so we had to make them ourselves, and this marks a long-term collaboration between myself and Professor John Brimacombe, who's now retired, in chemically synthesizing early biosynthetic intermediates of the pathway and analogues thereof to test substrate specificity and to find molecules that were not substrates but perhaps might even be inhibitors. And we made many dozens of variants on the theme here, and this just shows the retrosynthetic analysis of how you create these. You make an inositol derivative, a glucosamine derivative, and a phospholipid derivative, and basically bolt them back together again. So we made many such derivatives, and one of the key ones that we made is this one, and this is work that was done in my lab by Terry Smith at the time, where we were able finally to make a cell-permeable and selectively trypanosidal GPI inhibitor. Now, this is not a drug lead because it's far too big, it's got terrible pharmacokinetic properties, but it is a proof of concept chemical probe that was able to demonstrate trypanosidal activity in vitro, and gave us really evidence of some chemical validation that GPI biosynthesis is a potential drug target for African trypanosomiasis. The trick was actually caging the phosphate charge with this acetoxymethyl group. So that was nice. We could show that there's some chemical evidence for being able to selectively drug trypanosomes using this pathway. And of course, we also, whenever we're looking at potential drug targets, gain genetic validation of individual enzymes and steps to do that. And the way we like to do it in TRIPS, which are diploid organisms, so here, for example, are two copies of a gene that we think might be essential. So our preferred route, following the lead of George Cross and Christine Clayton, is to make conditional null mutants, where we knock out the 2 endogenous alleles and put in a tetracycline responsive ectopic copy. And then, of course, in the presence of tetracycline, they carry on living happily. And if you take the tetracycline away and they all die, then you have a genetic validation that gene target might be a useful target. So with these kind of studies, and many other, I hasten to add, basic biochemical studies from many laboratories, it was clear that we were identifying potential drug targets. But the question was, and this is, I'm talking about now late 90s and early 2000s, how do we exploit them? How do we take them forward? And I'm going to discuss that a little bit more later on. But if you'll forgive me, I'm going to give you a bit more basic science, because it is what we do. And I'm going to tell you about now some of the unique features of protein glycosylation in trypanosomes and how they also might be exploitable. So I'm going to tell you now not about GPI membrane anchors, which are these things at the bottom of the variant surface glycoproteins, or indeed anchoring the parasite's unique type of transferrin receptor, but I'm going to tell you about the carbohydrate chains which are known as Nglycans, the ones that are borne by asparagine residues, and they're these yellow glycans modeled onto the VSGs and the transferrin receptor. Now, my lab, under my supreme guidance, had studiously ignored N-glycosylation in this parasite for about 2 decades because I said it's not going to be very interesting because we know what the glycans are on VSGs and they're pretty ordinary. But actually, that turned out to be completely wrong, and there is something very funny about N-glycosylation. In trypanosomes, first of all, they make some huge final structures, and secondly, it turns out they have 2 simultaneously operating N-glycosylation pathways, which is totally unique in eukaryotic biology. So what about these giant glycans, first of all? Well, we knew for a long time that if you took a lectin like tomato lectin or ricin, which as well as being a weapon of mass destruction is a pretty useful research reagent, you could identify a whole smear of glycoproteins in the parasite. You can decorate this interesting fibrous mesh-like material that resides specifically inside the flagellar pocket of the parasite with those lectins as well. And you find that actually the whole of the endosomal lysosomal system, as well as the flagellar pocket, is very rich in these complex glycan recognizing lectins. And Abdullah Tree in my lab back in 2005 published a paper where we characterized some really unusual N-glycans, and Bob Haltwang is in the audience, so got another glycobiologist will be able to appreciate the unusualness of this. But on the sort of common core that all N-glycans have in all eukaryotes of 2 N-acetyl glucosamines and 3 mannose residues, they carry these colossal N-acetyl lactosamine repeat structures, which are really very large indeed, an average of 54 of these gal beta-1,4 glucnac repeats. So these are very, very large. They're not charged. And they have some very unusual structural features as well, which makes them actually very compact. And indeed, they're extremely gelatinous when you purify them. So this piqued our interest, the fact that these organisms were making, if you like, the world record of neutral N-glycans in size. And so we thought we'd better take a look at N-glycosylation after all. So I'm going to give you a moment of textbook revision on protein N-glycosylation. Protein N-glycosylation involves a thing called the dolichol cycle, where you start with a lipid in the endoplasmic reticulum membrane called dolichol phosphate, and sugars are added to it, 1 N-acetyl glucosamine 2, and then 5 mannose residues in this specific motif. That is then flipped into the lumen of the ER and completed to 9 mannose residues, and then 3 glucose residues are added. And this humongous structure is then transferred in a single en bloc step to glycosylation sequence in the nascent glycoproteins. And it's specifically this mature form that is transferred. So what about TRIPS? Well, they don't make the last set of those structures. They've dumped the genes that add those glucose residues from their genomes. And they do something really weird, which is they don't just use the biggest thing that they use to glycosylate proteins, but they also use this first competent species that appears in the lumen of the ER, this biantennary mannose 5 structure, and that is pretty unusual. So I won't go through the data, but I'll just try and summarize for you what we've learned about the glycosylation pathways over the years. And it turns out that there are basically these 2 parallel glycosylation pathways. So there's an oligosaccharyl transferase, which we call OST1, that takes this earliest dolichol-linked intermediate and transfers it to protein. And what happens to it once it's there? Well, it can get processed down through these what are called porcine-mannose structures, down to this very small mannose 3 structure. And then those structures can get built back up again into what are known as complex glycans, where you start to add N-acetyl glucosamine and galactose residues, all the way up to these huge polylacnac, these huge structures I just showed you a moment ago, where these will be extended off into infinity there. There's a second oligosaccharyl transferase, though, that takes the final mature intermediate that can be made and transfers that onto glycoproteins in the parasite. But those are locked into this little series here, which are known as oligomannose. They can only be processed, the smallest thing they can be processed to is mannose 5 glucnac 2, but you'll see that this mannose 5 structure is triantennary, has 3 arms, as opposed to this mannose 5 structure, which has 2 arms. So this locks it into this oligomannose series. Now, how does that compare with other eukaryotes, let's say mammals? Well, other mammals have a linear pathway. They transfer their dolichol intermediate with the 3 glucose residues on. They have an oligosaccharyl transferase system. They transfer that. the glucose residues are removed, they go through this oligomannose series, and they take the final product of oligomannose, and that is what they use to convert to the complex series. So it's a completely linear pathway, whereas the parasite has this dual mechanism. And I think the reason for this is that trypanosomes are really turbocharged. They're making 10 million VSG molecules every cell division to coat the surface. And I think they've evolved a system whereby they maximize the speed and efficiency of N-glycosylation by essentially having two simultaneous systems. Do we understand why, which glycosylation sites get the MAN5 and which glycosylation sites get the MAN9? Yes, we do. Work by Luis Izquierdo showed very nicely that we have two catalytic domain OST genes in the trypanosome genome, and the A gene is very selective for sequons, asparagine residues in acidic local environments, and they receive the MAN5 glucanac2 destined to become complex structures, and any other glycosylation sites left behind by A receive the mannose 9 and get locked into the oligomannose series. So actually we understand very well now the molecular details of N-glycosylation in the parasite. And we're particularly interested actually in seeing whether we can't exploit this oligosaccharide transferase system, which is very different actually from the mammalian one, as a potential therapeutic target in the future as well. So I've told you how the trypanosomes get to complex structures. They go through this route, and eventually they will make these enormous great structures that I introduced you before. But a really frustrating question is, well, which gene products are actually encoding the glycosyltransferase enzymes that make all this stuff? So that leaves us with a conundrum, which is that we have carbohydrate structures in search of the glycosyltransferases responsible for their biosynthesis. At the same time, in the genome, we do have glycosyltransferase genes in search of a function. So it seems like it's beholden to us to try and sort that out. So this slide summarizes all of the different glycosidic linkages that we've characterized in the parasite over the years, and there are about 40 of them altogether. And if you ask the question, how many of the genes do we know that perform the function to make these, the answer is not very many. So the ones that I've grayed out are all the glycosidic linkages in the parasite where we have no idea which gene is responsible for their formation. At the same time, we have a nice list of about 20 or so putative glycosyltransferases, so presumably some of these are making some of those. So we've had a very simple-minded program running for several years now, where we take one of the genes from the list, we make null and conditional null knockout mutants, we characterize the glycosylation phenotype, we see what effect that has on the parasite biology, and we publish it, and then we go and pick up another gene and go through the cycle over and over again. And it's a slow process, but thus far we've been able to do 4 such genes, and we've identified what's known as GNT1 and GNT2 from the parasite, as well as these 2 that are involved in decorating the GPI anchors, actually, of the procyclic form of the parasite. And I'd just like to acknowledge particularly Manuela Demaro's work on these 2, and Luis Izquierdo's work on these 2. But in doing so, we've developed the methodology, particularly mass spec-based methodologies, to do this rather more quickly. So we're hoping that we'll be able to go through the whole family over the next few years. So that's kind of work in progress, and some of those GTs are essential and therefore will also be potential drug targets. I'm going to finish off with a few minutes on some work that's very current, which is to do with nucleotide sugar biosynthesis. So glycosyltransferases are great, but they've got to have nucleotide sugars to power them. They're the high-energy donor molecules that the glycosyltransferases utilize to transfer the sugar to a growing sugar chain, for example, and tell you a little bit about nucleotide sugar biosynthesis in TRIPS. Nothing very unusual about it in terms of the pathways. TRIPS make GDP mannose, UDP glucanac, GDP fucose, UDP glucose, and UDP galactose by pretty conventional biosynthetic de novo biosynthesis routes. Nothing terribly surprising about that. It is actually a drug target rich area, because all of these gene knockouts that I show here, done in my lab, except for this one, which was done by my colleague Terry Smith in his own lab, all show essentiality, so they all have the potential to be therapeutic targets, so we're quite interested in them from that point of view. So much so that we've crystallized most of them in collaboration with colleagues like Bill Hunter and Dan Van Alten, and also with our friends in the Structural Genomics Consortium, Ray Huey and colleagues in Toronto. But the thing that's really rather weird about nucleotide sugar biosynthesis in TRIPS is where it takes place, because it takes place inside an organelle called the glycosome. Now, the glycosome is simply the trypanosome peroxisome, and this was an organelle wonderfully characterized by C.C. Wang and also Fred Opperdues and others. And glycosomes you can find in transmission electron micrograph are, as I said, the trypanosome peroxisomes, but they also contain all of the enzymes of glycolysis and are, to our surprise, all of the enzymes of nucleotide sugar biosynthesis, and that's unique. No other organism that we know of makes nucleotide sugars in their peroxisomes other than trypanosomes. That's all well and good, but it creates a problem for the parasite, which is if you make nucleotide sugars inside your peroxisome or glycosome, you've got to get them out into the cytosol, and actually from there they then need to get imported into the Golgi apparatus and the endoplasmic reticulum to do their jobs of work. But there are other nucleotide sugar transporters that are fully understood that do that, but we have no idea how the nucleotide sugars are going to get out of these membrane-bound organelles, the glycosomes, but we're assuming there's going to be a transmembrane transporter to do that. So having realized that, we thought, well, we should try and find out what it is, and in order to do that, we're going to need to determine the glycosome membrane proteome to see what is there. And we started then a few years ago now developing quantitative proteomics methods in trypanosomes, not rocket science, just simply transferring technologies that were being used in mammalian cell biology, for example, like SILAC proteomics, stable isotope labeling, and cell culture proteomics. And that yielded some very, very useful outputs in its own right, total proteomes of the bloodstream form and procyclic life cycle stages, and total phosphoproteomes of those two life cycle stages, which I think have created useful databases that I know many people refer to, so we're very happy about that. But the reason why we did it was really for ulterior motives, which was to be able to do this experiment, which was to determine a high-confidence glycosome proteome. And I'll just take you through the basics of this for those who are not so familiar with SILAC methodologies. We have trypanosomes, wild-type trypanosomes, labeled in medium with not isotopically labeled, so light, arginine, and lysine. And then we have identical trypanosomes which are transgenic, where we have tagged the glycosome membrane with green fluorescent protein, GFP. So we've taken one of the transmembrane proteins of the glycosome and tagged the outer surface of the glycosome with GFP. And these cells are labeled with heavy arginine and lysine, so they've all got heavy peptides, heavy proteins in them. We then take anti-GFP magnetic beads and pull out our transgenic GFP-labeled glycosomes. But the truth of proteomics, for those of you who do it, is a lot of rubbish comes along for the ride, especially in organelle pull-downs. So how can you tell what's real from rubbish? Well, everything that is real will have a highly enriched heavy isotope signature in its peptides, so very little blue ions and lots of red ions, whereas the garbage, stuff that just sticks equally to the beads from both of these, will have an isotope ratio of 1:1, and so you know that that's rubbish and you can ignore it. So how that looks in terms of data is each one of these crosses is an identified protein. So in this experiment we identified 2,300 proteins, the majority of which are absolute rubbish because they fall on this zero line of the isotope enrichment. And these ones, which are highly enriched, more than 8 standard deviations from the mean, are the authentic glycosomal proteins. And there's an intermediate group here, which are too technically complicated to bore you with, but some of which are also glycosomal. But within that family, we asked the question, well, how many of these are membrane proteins? And the ones with blue triangles are membrane proteins which span the bilayer more than once, in other words, which have the properties that one might associate with putative or potential transporters. So that got us down to only 32 glycosomal multipass membrane proteins, some of which we could remove from the list because they were already known and characterized, like the peroxins, which are involved in protein import into the glycosomes and so on and so forth, leaving us with 2 unknown function glycosomal multipass membrane proteins, Lucia did RNAi knockdowns of all 22. 6 of them gave a growth phenotype, which you'd expect, because if you starve the cell of sugar nucleotides, that's lethal. And 2 of those genes also gave a glycosylation phenotype when they were knocked down. So what she's been doing more recently is to try and functionally characterize those. And this is very preliminary data. I have to say none of this is published. But I wanted to put in something current into this award lecture. And so what she's done is use Xenopus oocytes to microinject RNA encoding one of those 2 putative sugar nucleotide transporter genes, and then got the protein expressed at the surface of the Xenopus oocyte, and then added radioactive nucleotide sugars to see whether the oocyte can then import them. And indeed, they can. So this the uptake of UDP galactose by those transformed oocytes, those microinjected oocytes. That's the water injection control. That's the injection of the mRNA for the transporter 1. And then we can compete that uptake with unlabeled nucleotide sugar as well. So it's looking quite good. We've got a long way to go to complete this work. But I'm very enthusiastic about this, because one of the great aims, I think, in biology is to annotate some of those hypothetical proteins of unknown function. In fact, I think we should give every PhD student a gene of hypothetical unknown function and say, go and figure out what that does, and then we'd know a bit more biochemistry and biology. But anyway, apart from that slightly flippant remark, I think this is a very nice example of functionally characterizing a new type of biochemical activity, and hopefully these parasite glycosome nucleotide sugar transporters may also be useful drug targets. Okay. So that brings me on to the final few minutes of the title of the talk, Translating the Trypanosome Surface for Therapeutics. Now, I'd love to stand here and say, okay, so I've shown you some targets that we've discovered and characterized over the years, and this is now where I show you how I've made those into a drug. And I can't, because although many of the drug targets discovered by my lab have been through the drug discovery process, none of them have made it to being a therapeutic. But because of the drive and desire to do that, it drove myself, and particularly my colleague Alan Fairlamb, to create a drug discovery capability, a translational engine, if you like, to translate not only our own targets, but those of our colleagues internationally, to try and see whether we could actually do something about making medicines for these horrible diseases. And that's what I'll tell you just a little bit about now. So we set up 10 years ago, gosh, time flies, 2006, a thing we call the Drug Discovery Unit in our university. And it operates a bit like a biotech company. It's not for profit, but it operates like a biotech company, but within a university setting. It's plugged into extensive academic expertise and technology platforms, so it actually has resources not available to most biotech companies. It's doing quite well. Its current funding streams total about £33 million, so it has a burn rate of over £6 million a year. It's got biopharma industry drug discovery experience team, 85 people, 660 collective years of experience in biopharma. So this is not academics pretending or trying to do drug discovery. This is actually bringing drug discovery professionals into the academic center to do the drug discovery in-house. And our colleagues come from lots of pharma companies like GSK, AstraZeneca, Merck, Pfizer, etc. So what we're trying to do is combine excellence in basic science with biopharma industry expertise. It's not in competition with the pharma industry at all. It's trying to complement it. We work on diseases of the developing world, both tropical and orphan diseases, and we work on diseases which are not just neglected tropical diseases, but also things like Alzheimer's disease, eczema, cystic fibrosis, and so on, where we can take innovative targets from other academic colleagues and identify new approaches and take those innovative targets through early stage drug discovery, and then we partner with them with industrial partners downstream. So that's really the model. It's to do two things: to advance drug discovery in neglected diseases, and also to advance innovative targets towards partnering with industry. Now, although Alan and I set the DDU up, I'd like to just emphasize that it's run by a management team. Paul Wyatt is the head of the drug discovery unit, and Ian Gilbert is head of medicinal chemistry. Kevin Reed is head of pharmacokinetics, distribution metabolism of pharmacokinetics, DMPK. David Gray is head of biology. That's all the screening platforms. We have a business development manager who liaises with all our industrial collaborators, and Andrew Woodland project manages the innovative targets which are out with the tropical disease work. So something like this couldn't operate without a really top quality professional management team that Alan and I do our best not to interfere with, which is really important. So I think it's a pretty useful organization. It's got all of the things you might consider you need to do drug discovery. We've got a very good target selection system where we purge projects of wishful thinking and look at what's their level of validation, what's the evidence of drugability, what's assay feasibility is the most important thing. Actually, no assay, no drug discovery program. So that's often the key one, and so on and so forth. We have lots of robotics for doing screens. We've got very, very good compound libraries. We've got very good data management and informatics, obviously computational chemistry, structural biology that we use as well, in vitro and in vivo models of disease, and perhaps uniquely in a university setting, a really sophisticated pharmacokinetics group who are able to, and we bring in toxicology and pharmacokinetics very early into our drug discovery pipelines. So with that kind of capability, what do we do? So I'm going to give you two incredibly short vignettes of a couple of examples of drug discovery campaigns. You can do drug discovery campaigns starting with an individual molecular target. And the last talk today by Meg Phillips is going to be an absolutely beautiful example of that. And you can then get hit molecules against that target and develop them towards preclinical candidates. Or you can do a phenotypic screen where you start with a cell-based screen, so in our case, killing microbes, find molecules that kill microbes and get the cell-based hits and then figure out what their targets are later, but again, develop them through to preclinical candidates. I'm going to show you one example of a target-based screen and one example of a cell-based screen, but it's going to be super quick. So the first one was where we targeted a specific enzyme called N-myristoyl transferase. This was a collaboration with Debbie Smith from York University, who brought the target to us, and also our colleagues in SGC, Structural Genomics Consortium Toronto, that solved the first crystal structure in collaboration with us on this target. And this was published some years ago now, 2010, in Nature. So how does a target-based program work? You perform a high-throughput screen. This was the best hit that we got, not particularly potent. It was only 20 micromolar against the parasite N-myristoyl transferase. But through rounds of medicinal chemistry, the team was able to improve that to 2 nanomolar. So that's a 10,000-fold jump in potency. But more important, actually, than the potency is this molecule has very good solubility and pharmacokinetic properties. We know that it engages the target. Here's the molecule embedded in the crystal structure of the N-myristoyl transferase enzyme itself. And so we had a molecule which was very trypanocidal, rapidly trypanocidal. It actually killed trypanosomes in an interesting way, making them get this big eye phenotype, first characterized by Mark Field in his clathrin knockdowns. It works on target. The potency against the molecular target and the potency against the whole parasite track very well together, and it has very good pharmacokinetic properties. Most importantly, it cures the animal model of the Trypanosomiasis disease extremely well at 10 mg per gig twice a day. So, great stuff, lovely project, nice paper, failed completely as a therapeutic. Not because it isn't a good molecule, but because it's a clinical candidate for stage 1 disease when the parasite is in the blood, but not for the stage 2 disease of the parasite, stage 2 of the disease when it gets into the brain. And almost the same day that we generated this molecule, DNDi, who's the downstream international partner who will take molecules into clinical trials, declared that they were no longer interested in stage 1 only drug candidates. So it's parked for that reason. And I just throw that one in because it's a great example of how all the science can go really well and then you can be scuppered by the harsh realities of drug development. And CC is nodding from his Merck Sharp and Dome experiences. I've seen that many times. So very briefly, an example of something that starts phenotypically from a cell-based screen. This is a project which was in malaria, and a huge number of co-authors. This was published in an article in Nature just last year. I'm not on this paper, incidentally, so I can be particularly proud of this one. Ian Gilbert and Kevin Reed from the DDU directed this, and this is the small team in Dundee actually that did all of the chemistry on creating this molecule. Huge number of other collaborators because this was coordinated through Medicines for Malaria venture in Geneva and brought in a lot of expertise to figure out the mode of action of the molecule at the end of the day. So this was a very small screen that was run against the blood stage of Plasmodium falciparum, the malaria parasite, and actually this was one of the original hits that came out of that screen. It came out of a kinase library screen. Only 5,000 compounds were screened. This is one of the hits. So it was reasonably potent, 120 nanomolar, but it had terrible clearance properties in animals, and so we started to do some minor optimizations and took a bit of hit on potency to begin with. It was still bad for clearance, but the solubility was so bad here we couldn't measure it. Solubility started to improve, a few more modifications. Again, taking the hit of losing potency now only 700 nanomolar, but now the properties in terms of clearance and solubility and lipophilicity are looking much, much better. And then finally making a modification to improve the solubility still more and reduce the plasma protein binding capacity. And we ended up pulling back the potency to 50 nanomolar. And this compound, known as 498, was declared a preclinical candidate by Medicines for Malaria venture and is being taken forward now in partnership with Merck Serono, who are paying for the preclinical development and will put it into phase 1 clinical trials, hopefully very early next year. So that's a phenotypic screen. The intellectually frustrating thing is you don't know what the molecular target or targets are that it's engaging with. And this was sorted out by very nice collaboration with Julian Rayner at the Sanger Institute in England, and also David Fiddick here in the States. And basically we raised resistant mutants of the parasite to the drug, and all the mutations mapped to eukaryotic translation elongation factor 2, telling us that this is actually a protein synthesis inhibitor, a very potent one, very selective. Although these are highly conserved proteins across eukaryotic evolution, this molecule only hits the plasmodium EF2 and not the human one. Now, we started from a kinase screen, and we ended up with a protein synthesis inhibitor, which just goes to show there's no substitute for activity. It's a very nice molecule, 498, and it's, as I say, it's in development through Medicines for Malaria venture, and because it's a protein synthesis inhibitor, actually it affects every single life cycle stage of this parasite, which means it not only has the right properties to cure malaria, but also to act as a chemopreventative, to prevent infection, and also nicely, because it affects the sexual stages of the parasite as well, it also has the right properties to give transmission blocking. One of the problems with a lot of therapeutics in the malaria area is you give the drug to a patient, you cure their infection, they feel better, but they remain infectious to mosquitoes for maybe 2 weeks, and they can pass it on to all of the people around them. So this drug will actually has the potential to sort that out. So we're hopeful that will progress towards the clinic. So that's the DDU. I told you about the human trypanosomiasis candidate that got parked. I've told you about the malaria preclinical candidate that's doing very well with Merck Serono. We just declared, literally a couple of weeks ago, a new candidate for visceral leishmaniasis, which I'm not going to tell you about for time. We have 3 compound series which are in lead optimization for Chagas disease, for T. cruzi. And Paul Wyatt also runs a very powerful group working on TB drug optimization, and he's hoping to be able to declare a clinical candidate, new clinical candidate for TB later on this year. So we think it's going pretty well. And what we're trying to do really in Dundee Life Sciences is just to help translate academic innovation, taking innovative academic biology, whether it's parasitology or otherwise, let it see professional early stage drug discovery, and then partner the outputs with product development partnerships like MMV and so on, or with pharma partners to get drug development and through to clinical trials. So it's an interesting activity, and one that I think actually sits really well alongside basic science. This is not to replace basic science, it's to have a translational engine in place that when there's opportunity, there are no barriers to advancing things through early stage drug discovery to get to a point where you're having an appropriate conversation with pharmaceutical partners. I'm going to skip that for the interest of time. That's just some work that we're doing also on diagnostics. But I just wrap up by thanking my mentors, because it's important when you get an award like this, which is really a wonderful honour, to remember how you got here and who helped you. So George Cross was my postdoctoral supervisor. Peter Overath was doing a sabbatical in his lab when I was doing that and taught me how to do experiments properly. Raymond Dweck was my postdoctoral supervisor for my second postdoc when we completed the GPI anchor structure. And Phil Cohen recruited me to Dundee and taught me that you can dream large and try and do bigger things outside of your own laboratory interests. And I'd particularly like to thank Alan Fairlammer, who I think nominated me for this award and is my partner in translational crime and co-founder of the Drug Discovery Unit. And I'd also like to thank our principal funder, the Wellcome Trust. Thank you very much.